I'd like to introduce this evening's speakers who will be with us. First, we will hear from Dr. Scott Rifkin, Ph.D. He's an associate professor of ecology, behavior, and evolution at UC San Diego. He's also an alum of the RSI program I mentioned. He participated in 1992. So I'm going to go ahead and turn this over to Dr. Rifkin to begin. He has requested that for a moment, if you could all turn on your videos, if you have them, and your audio, if you have it available, and we're all just going to say a big hello. He'd like to see your smiling faces and kind of catch a glimpse of everyone that he's going to be speaking to this evening. Hi, everyone. I'm glad to see you as well. Lots of folks. Hi, good evening. How's everyone's school year going? Thumbs up, thumbs down, to the side?
Yeah, pretty good.
Thank you and welcome. Also, throughout his presentation, Dr. Rifkin may ask you to turn on your audio or video again. He does have some interactive portions. That said, take it away, Dr. Rifkin. You may turn off your video now. Thank you.
Okay, so I'm interested in variation and how biological variation is generated and structured. And so what I'd like to do today is first tell you a little bit of a research story that gives you some of the ideas of the kinds of questions that I work on in this area and some of the questions that are out there. And I'll follow that with a little interactive portion about variation in society that I do in one of my courses. And then touch on some, I guess, key science-related skills that I think students, well, key science-related skills that students probably should need, even if they don't go on to be scientists. All right, so I'll start with the research part of it. And again, and also if you have questions, please do type them in the chat. There's going to be a question period afterwards. All right, so obviously variation is a pervasive feature of life. It's the defining feature of life. There's tons of variation out there, and this is a lot of the reason why, oftentimes the reason why biologists go on to become biologists, because they're fascinated by this variation and how it came to be. And of course, here we have variation between species, but there's variation within species as well. So here's a lot of variation in ladybugs that you see on the branch. Now, even though there's a ton of variation out there, there's some variation that we don't see. So some variation is not allowed. So for example, even though it would be really awesome to have wings, vertebrates just don't have 6 limbs. So you're never going to see a Pegasus. It'd be very hard for us to grow wings out of our back. It just doesn't happen. And this is actually an interesting question. Why do we have some variation that's allowed and some variation that's not allowed? And this is related to the question of how we get the variation in the first place. So my research program is really focused on this question. How do you get variation in the first place? How do you get differences between organisms? Why are they different? And we can sort of chalk it up to these different influences. We have environmental differences between the way organisms grow up. We have genetic differences. And we have randomness. And all of these funnel through development to produce the variation that we see. Now, that's sort of a descriptive question, you know, how are they different? My lab tends to focus on developmental mechanisms. So how do they get that way? How do these environmental, genetic, and random differences, how are they filtered through the developmental process to actually produce variation? You might imagine that there might be some differences on the left side of this that are filtered through the development and just don't make a difference. They just don't do anything. There might be some that you have a little change in the genetics and boom, you have a huge difference, right? So the developmental mechanisms are key to taking that variable causes and producing some outcome, whether variable or not. Now, when we talk about developmental mechanisms, at least for this talk and a lot of my work, I'm going to predominantly focus on gene expression. So what gene expression is you have your DNA in every cell, but that DNA is inert. It needs to be read. it needs to be read by your cells to produce a molecule called RNA. RNA is then translated into protein, and protein is the things that does things in the cell. It's not entirely true. We now know that there actually are active molecules of RNA that do things as well. At least the standard paradigm is you have DNA, it's read into RNA, RNA makes protein, and protein is really the workhorse of the cell. And this process is called gene expression. Gene expression is how much the DNA is read. The nice thing about gene expression, especially nowadays, is that there's tons of technologies for measuring it. So we can really measure variation, measure gene expression. And if we want to talk about variation, we really need to have a quantitative measure of it. That's crucial. And increasingly, being able to quantitate things in biology is just a growing part of the field. So what I'm going to talk about is a story that has to do with how you get variation on the left side filtered through gene expression to produce differences between organisms. Here's an example. So here are a bunch of E. coli. These are bacterial cells. They live in your gut. They also live on lab plates, plates in the lab. And about 6 hours before this picture was taken, there was one. Okay? They divided, they clonally propagated, and they had a green fluorescent protein in them that fluoresced at some level. Now, all of these bacteria are genetically identical. They're all right next to each other, and yet you can see that they're different. Right? They're expressing this gene at different levels. Here's another example. So here's a bunch of pea plants. Now, what these pea plants have is they have a mutation in a particular gene. And these are all, again, genetically identical. They're from the same plant, but each of these, but you see that there's variation in the pods. And so even though they have a particular mutation, the effect of that mutation is variable. And this is called partial penetrance. It doesn't, the effect of mutation doesn't completely penetrate the whole organism, or all the individuals. So I'm going to tell you a story about partial penetrance. And again, this is a story about how randomness affects gene expression to differences between organisms. So in order to do this, in order to really study this, we need to pick a system where we can isolate the effects of randomness. Okay, so a lot of what scientists do is, and especially biologists, is sort of try to pick the right organism to answer the question you're interested in. The organism has to have particular characteristics. So in this case, we want to be able to limit the genetic differences, ideally eliminate them, limit the environmental differences so that the only causal differences are randomness. So we're going to do this with a species of nematode called C. elegans, very common very common model organism. What's nice about this is that they're almost all hermaphroditic. So there are two sexes. There are males. They're about 0.5% of these worms are males, and nearly all the rest are hermaphroditic. That means they can self-fertilize, and that's what they do most of the time. That means they're very inbred. So that means that there's not much genetic variation. Now we can grow them together on plates where we've effectively eliminated environmental variation. So if there are differences between these worms that we can observe, then it's really just the only thing left is just random differences. So I'm going to focus on early development in these worms. And what's nice about these worms is that they have a very stereotyped development. So for example, in you or me, if I look at a particular cell in my hand and then look at the exact same position in your hand, those cells would have very different histories. That's just the way mammals develop. These worms are very different. These worms have a very, very stereotyped pattern of development. So if I take one of these cells in these embryos in one worm, and I take the same cell in another worm, I know that actually they have the same exact pattern of cell division, nearly the exact same timing of cell division. And so we can actually map out that cell lineage, and this cell lineage, these are charting cell divisions over time from top to bottom. And this is going to be basically the same timing. So time is on the y-axis. This is going to be the same timing across these worms. We're going to look at this particular cell lineage, the E cell lineage. And the reason for that is that these cells, the E cells, become the intestine. So all these 20 descendants of the E cells become the intestine. And just as a picture of what that is, you can see that's here. So the arrows point to where the E cell is and when it divides. Okay, now I told you to talk about gene expression. So there is, the way that gene expression works is you have these molecules called transcription factors, the proteins, and they bind to the DNA and basically activate the expression of other genes. So they bind in front of a gene and activate the expression. They're very specific, right? They don't bind willy-nilly over the genome. They bind in front of specific genes and activate the next gene. And so I have this network here, and what the arrows are actually these interactions, these regulatory interactions. So all of the genes here are transcription factors, and this SKN1 binds right in front of the MED1/2 genes and activates it. It also binds in front of the N3 and then activates that. N3 activates N1 and so on. Okay, so this is a genetic network. And the goal is to activate this downstream gene in green here called L2. If you get that on, it stays on throughout the worm's life and coordinates the development of the intestine. It basically then binds to like 1,000 other genes that then build the intestine and make sure it works and all that. So the goal for the worm is in a very short period of time, basically start with skin 1, activate these other genes, and then activate L2, and then it's home free. That's the goal. Now, it turns out we can measure the expression of these genes. So what I'm showing here are some of these embryos. Nuclei are stained in blue. And the green are actually individual molecules of RNA. So individual molecules of this L2 RNA. And you can see the intestine is sort of in the middle of the embryos. Okay? So that's what normal gene expression looks like at particular stages in development. Okay, and in fact, we can actually measure all the genes in this network. So here's a picture in the upper left, that's staining nuclei. In the upper right, that's this N3 gene. In the lower right, there's actually 2 genes. There's MED1 and 2 is sort of expressed early, and L2 is expressed later. And then in the lower left, there's this N1 gene. And you can see that the timing of expression of these genes differs in the different stages, right? In the upper left, you can see there's some early embryos with very few cells. There's some later embryos with more cells. And so we can get essentially a time course of development this way, where we measure how many dots there are, and we count the nuclei, and we know then how much any particular gene is being expressed at any particular time. So we can generate a plot that looks like this. So this is what wild type expression looks like. If these MED1 and 2 is expressed, N3 is expressed a little later, N1 is expressed a little later and for a long period of time, and then L2 is expressed and stays on throughout the worm's life. Okay, so now what we want to do is we want to mess this up. So what we're going to do is we're going to mutate the top gene in this network. And look what happens to the bottom. Right? So when we do this, if we just look at the outcomes, you can see that I just want you to focus on one of these lines. Wild type is just your normal ones, and 100% of embryos in a wild type worm, not a mutant, will develop intestinal cells. But if you make one of these mutations, then you get variable numbers. Some of them develop intestines, some of them don't. So I want to try to figure out how that happens. So what we did was here's a picture of this L2 gene, the downstream gene in one of these mutants. Now, these mutants are all genetically identical, and yet you see that some of them express L2, those that were the middle one that has all the pink, those are going to have intestines, and some of them don't. Those are not going to have intestines. So when we look at gene expression in this, we can see that in the mutant, on the left is wild type, on the right is the mutant, and you can see that expression has changed. And I want you to focus on the purple and the green. So the green is this downstream gene, L2. You're basically trying to get that expressed. And you can see that the purple has a really nice curve in the wild type, and then the mutant is highly variable. So this brings up a hypothesis. The hypothesis is that there's some threshold level of that purple gene, that N1, that you need to get to in order to activate the downstream gene, L2, the green gene. So here's a picture of what that looked like. This is normal times. You're always above that threshold. And with the mutant, you fall below the threshold. And so we were able to show that in fact that is the case. So if you just focus on the green on the block here, the wild type, the normal ones are in the blue dots. This is basically on the x-axis is the upstream gene, on the y-axis the downstream gene. You see that on the blue, normally you have lots of the downstream gene, you get lots of the upstream gene. In the red, you get lower levels of the upstream gene, leading to sometimes you express the downstream gene, sometimes you don't. Okay, so I think, so we were able to track it down and figure out that this partial penetrance is due to randomness, it turns out, in the expression of this upstream gene. All right, so that's what I want to talk about for that. That sort of gives you a flavor of this work, and I can answer questions after the talk. But I want to do a quick. I want to sort of switch gears now. So that's a sort of flavor of some of the interesting questions, at least that, well, I think they're interesting, that my lab works on. But I want to sort of switch to some topics about science in general, modern science in general, and some ideas that I'd like you to think about as you're structuring your teaching. Some of you are biologists, some of you I imagine are teach other scientific subjects. So I'll make for the biologists, I want to do for everybody, I want to do a quick exercise showing how you can how incorporating the modern biology into modern biological findings into teaching genetics, or genetics at a high school level or even lower levels, can really address perhaps some societal problems that we have. Okay, so here's the exercise. So here's a picture of human genetic variation. Okay? And the thing is, the reason I bring this up is because a lot of times, at least both in college biology and I think in secondary school and lower biology, we tend to shy away from talking about humans. You know, we talk about Mendel's P's, we talk about other sorts of examples for genetics, but we sort of shy away from about humans. And this has some unfortunate consequences that I hope I'll be able to illustrate. So this is what human genetic variation looks like. So basically, all the purple is DNA that makes us humans, basically shared by everyone. There's no variation in humans. And that's 99.9% of our DNA. And then 0.1% of our DNA makes humans different from each other. And it's not shared by everyone. And so that little 0.1%, we have lots of variation. Okay, so what I'd like you to do is the following. I'm going to set up a way to think about human genetic variation. So it's just a very simple model for it. And then I'll ask you to do an exercise. So when we if we take a group of humans, we can represent the amount of variation by a circle. Okay, so there's a group of people, they're genetically different from each other in that little 0.1%. And if there's a lot of variation between them, then it's a big circle. If there's a small amount of variation, it's a smaller circle. Okay. Now, if we have two groups, then we can actually represent we can look at how they relate to each other. So if they share a lot of the variation, then the two circles will overlap. However, if it could be that one group is genetically variable within itself, but the places where it's variable is very different from the other group. And so then you get the lower part where they don't overlap so much. Okay. So the upper part, they actually have a lot of overlapping genetic variation. The lower part, they don't have much. They're actually genetically distinct from each other, even though within a group there might be some variation. Okay. And that little overlap is the sort of shared within group variation. Okay, so what I'd like you to do is the following. I'd like you to draw, using that Venn diagram picture, those circle model, I'd like you to sketch out on a piece of paper what you think the picture would look like for human racial variation. Now, I don't want to get into here definitions of race. That's a very difficult question. In fact, this exercise addresses it. So I'm just going to use US Census categories. So what I'd like you to do is draw a picture of how genetic variation looks like between white Americans, African Americans, and Asian Americans. Okay, so those are going to be your three circles. And then, so I want you to think about how much genetic variation is, that's the size of the circles, and then how much overlap there is between the groups in their genetic variation, that's the overlap of the circles. So take a moment, right, take like a minute, and just sketch out what that looks like. If there's any questions about how to do it, if it wasn't clear, then you can, just unmute yourself and ask. Okay, hopefully you've drawn a picture. What I'll ask is for some volunteers, if you want to volunteer, what I just like to say, I'd like to see is just hold up your drawing in front of the camera, if that's okay. Okay, and
I'll just step in for a moment. Anyone who would like to, as he says, feel free to turn on your camera and hold up your drawing.
I think I might stop share.
Perhaps you can call on folks or speak to folks if you want to speak about what you have. This is mine.
Okay. Can people see Eric's and Molly's?
Yes.
Or is my camera messing? Is
I
my
see
sharing screen?
Eric's. I think I saw Lisa hold one up. Anyone else? If I'm pronouncing this correctly, Pachon, Diana. If you're unable to use your video, I understand. Happy Hollow is a name I see.
Okay, so hopefully being able to
I see a lot of intersections there.
Oh yeah.
Ms. Smith.
Got one.
Yes, I see quite a few.
Okay, so thank you. So let me.
And anyone else can go ahead. Adrian, were you about to share with us? Let's see yours. If you'd like to. I think it's taking the shape of your background. Oh no. That's okay.
Okay,
right.
so let me talk about why I do this. So when you do this with, what I've done in my class with college students, and when people have done this with secondary school students and lower grade students, what the most common thing that people draw is three circles that overlap a little bit. Now, what that means is that people are conceiving of races as genetically distinct. Some of you, I couldn't see all the pictures, but some of you may have drawn that, some of you didn't. But that's the conception, that there's a lot of genetic distinctness to different races. So here's the current scientific consensus of what it looks like. Okay, this is actually a little bit exaggerated. It's actually probably a little bit more overlapping. So the scientific consensus from, when we do this, when we sequence, you can now sequence whole genomes. When you sequence whole genomes and you say, okay, let's just take the US Census categories and see how much variation there is. The vast majority of variation in humans is just between two individuals, regardless of what we call races. What that means is that these racial categories are not, the genetic basis of a racial category is actually quite weak. And this is actually a surprise to a lot of students. Now, in order to get to this, right, you imagine there's actually a lot of genetics you can teach to get to this point. So you can teach genetics sort of leading up to this point, and then you can teach genetics coming off of this point. But it's an inherently interesting question to kids, right? How different are we? And it's also very important societally, because it shows that this race concept doesn't really have much of a biological basis. And that's something that I think is actually very crucial to instill right now, and to instill what is our best understanding of human variation at this current point in time, at this current scientific point in time. So I think that's a fruitful way for those of you who teach genetics to think about incorporating human genetic variation into the way you teach about variation. Now, there's actually a group, this BSCS science learning that you can see on the bottom of the slide. There's a group that actually puts out a curriculum for doing this for secondary school students. Actually, I think they have a K through 12 curriculum. It varies depending on the age, but they have materials that you can use to work through this idea about the structure of human genetic variation. So BSCS, if you're interested, you can look it up, and they're a pleasure to work with. Okay, so that's sort of what I want to talk about a suggestion for genetics. And the last thing I'll say is the following. For those, for students of yours who are really interested in science, and particularly those who are really interested in biology, I teach biology, and it's my experience that a lot of students in come to biology, not all of them, but a lot of students come to biology because they like science, but they don't like math. They don't want to do physics, too much math. Don't want to do, chemistry. They don't want to do math, too much math. I think that's a problem. And the reason that's a problem is because every scientific field now really does two things. There's a lot of math and a lot of computer science. And so it's good, I think, to incorporate quantitation into biology teaching to show that, in fact, there's a lot of that this is an important skill. You really want to understand the biological world, important skill. I also think it's important for students who might like physics, for example, to use biology examples to show that these disciplines are really disciplinary boundaries are a little artificial. For example, I think it's much more interesting, I do, but I'm a biologist, and physics people may disagree, or engineers might disagree, but I think it's a lot more interesting to think about teaching mechanical advantage or torque forces by thinking about the forces that our muscles have to generate to chew, right? And then if you think about that, now when humans started cooking, and back a long time ago, when they started cooking, then all of a sudden the forces they needed to chew were lessened because the food was softer. So what does that mean? It turns out that there's a theory that cooking, softening food, meant that you had your muscles that you came down from the top of your head down to your jaw to be able to actually crunch hard foods. Well, those muscles didn't need to be as big. That allowed more room for the head to expand and for the brain to expand. So it's thought that basically this mechanical property, lowered mechanical requirements for the muscles in the head, actually led to the expansion of the brain, and it was an important force in human evolution. That sort of context, I think, makes it a lot more fun to learn the physics, and also shows that these fields are very interrelated, and that you can't really answer that question about how human skulls got bigger and how they evolved without really understanding something about the physics and doing models like that. The last thing I'll say is that another aspect of biology that's really crucial, and it's not just biology, I think it's actually just for citizenship, is to really understand numbers. And what I mean by that is that our society is really awash in numbers right now. And there's this idea that if you put a number to something, then it gives it rigor, right, and scientific backing. And so you look on the TV and people are always quoting numbers and studies at you in every situation. And the thing is that many of these numbers and many of these studies are just nonsense. They're not very useful. And so I think a key skill that students need to learn is essentially, well, skepticism, that's sort of a key attitude, but also just knowing what sort of questions you would need to answer in order to determine that this number that this person is spouting at you is valid. Now, when I'm watching TV and I hear a number, I don't necessarily know that that study is good or bad. But the thing is that I know what questions I would need to ask. I think that's the important skill. When someone's talking about something causing something, knowing about what does it take to distinguish correlation from causation, that sort of skill. So that's sort of, I mean, that's sort of applied statistics, but it's not like how to do a t-test. It's what are the concepts in statistics that allow you to evaluate, essentially, what do you need to know in order to satisfy your skepticism, in order to know whether someone is being, whether these numbers are good or not. And there's actually a book that just came out called Calling Bullshit by a professor, two professors up at the University of Washington, which is really good and sort of dedicated to this idea. Like, what are the questions you need to ask in order to distinguish, what are the questions you need to ask that would need to be answered in order for you to know whether these numbers are valid or not, and not just take them at face value. All right, so I will stop there. And if there are a couple of questions, we can.
So I'm back with you now. First question, please. Does the size of the circles in the image you presented indicate equal variation within each race?
Okay, so that's a great question. Now, so the answer to that is, yeah, they're not exactly the same size. So there is more variation within African Americans. And the reason for that is that humans evolved in Africa. And it took a long time for humans to come out of Africa. And so there was time to build up human variation. Now, on the other hand, a lot of African Americans are descended from slaves who came from West Africa. So that was just a subset of African variation. Not all African Americans, obviously. So the African American circle should be a little bit bigger, but just a little bit bigger. It's really not that much bigger. So yeah, it's hard to see with circles exactly what size they are. Is the BS human genetics free? So I don't know whether it's free. I think it is, but that's something definitely that they'd be able to do. I think it is available. Okay. I'm not sure though. I can't.
Okay. Kim said she's happy to do some of that research as well. The next question, so that we can have it in the recording as well. Is it true that because human skulls got smaller through evolution, our nasal cavities diminished in size, and that's why there are so many people that snore and have apnea? And I would like to add for myself, I inherited narrow nasal passages from my father. My allergies are extreme. So I'd love to hear the answer to that one.
So the answer to the quiet one is I don't actually know. I will say that it is that what I'm pretty sure is true is that one of the reasons you have to have your wisdom teeth, often have to have your wisdom teeth extracted, is because in fact our jaws have gotten shorter, right? And so you just don't have space there. Yeah. In part, possibly because we don't need those teeth there for chewing so much anymore, right? But I don't know this question about the human skulls. I mean, I don't know the question about the nasal passages and that, but somebody will have studied it.
We can try to do some research on our end as well and get back to you, Diana. We'll see what we can find. Or yeah. If, as I used to tell my students, if you find it first, let us know and we'll get a few bonus points.
Yeah, and I wonder if it also has to do with, well, yeah, I don't know the answer.
We have one further question that came in. In a book, she read it in a book called Breathe. So there may be something behind that, or seems to be. Very interesting, thank you. A question that came in privately, what kind of food will increase brain function? We hear a lot about superfoods. What can you recommend?
Okay. So I used to teach, I taught introductory statistics for about 7 years at UCSD. Partly because, as I mentioned, I actually think that, I wanted my students to come away skeptical about numbers. I think that's the single most important thing that they would learn at college. But I also loved to give examples from nutritional epidemiology, basically what foods you eat, because the field is utter junk, okay? And it's utter junk because it's really hard to do this sort of thing in humans. It's hard to distinguish correlation from causation. It's very hard to have like normal people control what they eat, control their diets. You can't do an experiment. And so, for example, you know, the newspapers love to report these associations between, say, red meat and you know, heart disease, red meat, and all this stuff. I don't eat red meat. I think it'd be great if people don't. But the thing is that they report these studies from some of these just observational studies. So these are studies that are not experiments; they're just observing things. And then they try to go back and actually validate them by controlling what people eat. And it turns out that of something like 100 studies that have been published and widely advertised, none were actually confirmed when you actually do the experiment. So there's some great literature about that particular field. And of course, it's very sexy and very popular in popular press. But as far as a scientific field, it's really just garbage. So sorry, I don't know.
Just letting me know we have time for one more question.
Okay.
But I love the breadth of your answers. Not only entertaining, but also very informative. So thank you. You discuss with students the fact that humans are visual mammals and visual variation is overemphasized throughout history. I'm curious why humans emphasize visual differences so much. What's the evolutionary basis for this in humans? And as they say on the news, 30 seconds, please. No.
So I was actually an anthropology major in college and studied biological anthropology. I studied primate behavior and evolution. And primates are visual mammals. Like we do things with our eyes, unlike others. And so it's a really great point, especially when we talk about human differences, because that's what we see, right? We don't see all the other stuff. We see that. And so part of the reason is just because we're primates. We evolved from primates, and primates had developed large eyes, and that was a really key part of primate evolution. But I think it's really important, as you note, that that's sort of biasing what we see and what we perceive that we see, those differences. We're very good at those. That's not necessarily general about every other part of human anatomy and human physiology. Right? So it may be, like, as you say, visual cues are probably overrepresented in the differences between things.
That makes a lot of sense. And thank you very much for that information and kind of that explanation of, again, that perhaps what seemed a seemingly overemphasis on that aspect of our evolution. And with that, I'm going to thank you so much again. I'm sure we'd all give you a big round of applause. So if Scott could join us once more for just a couple of minutes, he would like to speak to the educators about some opportunities for your classrooms. Yeah,
so I just want to say that if, especially if you're in the San Diego area and would like teach a biology class in the San Diego area, or maybe one of these other classes. My lab does a bunch of outreach activities. And so if you're interested, we could please definitely contact me. We're always looking for classrooms to go to. And the other thing I wanted to say is that in the past, we've also gone into classrooms just to talk about science as a career and what's, you know, what scientists do. And so that could be done online. And so if you know, so again, just contact me if you're interested in either of those.
Wonderful. Thank you so much.